No, I know it's partners because
So now,
and from the Germany for each. Yeah. That's really for our father. We'll go to because as well. Did have this issue. Was it? What 2 weeks?
Je vais vous en chercher un tout à l'heure.
Good morning, everybody. Thank you for being present. And we have today the Séance du Président, which is devoted to endometrium, endometriosis, and adenomyosis. And we have people also online.
With the microphone is better. Yes, we have people following us online. Thank you, Mr. President, Professor, Doctor, Mesdames, Messieurs, bonjour. Professor, doctors, ladies and gentlemen, thank you for being here. Comme toujours, 10 minutes seront consacrées aux échanges avec l'orateur à la fin de chaque conférence. Vous le savez, les membres de la compagnie peuvent poser leurs questions via Zoom, en direct ou via le chat. N'hésitez pas à vous signaler. Si vous nous suivez sur YouTube, vous pouvez également nous envoyer vos questions via le chat et n'oubliez pas de préciser vos noms, prénoms et institutions. Crucial process in the conception cycle, the decidual reaction, can be weakened by miscarriage. As Professor Brosens is going to tell us, a recent study even shows that prior miscarriages disrupt the inner cycle homeostasis of the endometrium, impairing the decidual reaction in the following cycle. Mr. President, you are going to introduce this research partner from the UK.
Yes, Jan Brosens is a Belgian gynecologist. He graduated from KU Leuven in Belgium in 1990 and pursued postgraduate training in obstetrics and gynecology in the United Kingdom. He became a member of the Royal College of Obstetricians and Gynecologists in 1995 and a fellow in 2008. He obtained a PhD from the University of London in 1999 and was awarded a Wellcome Trust Clinical Scientist Fellowship in 1998, became a Wellcome Trust Investigator in 2018. He joined Imperial College London, first as Chair of Reproductive Sciences and then as Chair of Reproductive Medicine, and currently he is Professor of Obstetrics and Gynecology at the University of Warwick. He leads the Implantation Research Clinic at University Hospitals Coventry and Warwickshire, and is the scientific director of Tommy's National Miscarriage Research Centre, a partnership between Imperial College London, University of Birmingham, University of Warwick, and the affiliated NHS Trust. He is a world specialist of decidualization, particular differentiation of endometrial stromal cells that is thought to be needed for appropriate implantation of the blastocyst.
Thank you very much. Mr. President, it's a true privilege for me as, being Belgian, but for the last 34 years working in the UK, to be here today and to give this talk. So thank you so much for the invitation. Before I start, some disclosures. So some of the biomarkers I'm going to discuss have been patented by the University of Warwick. But I really want to start almost more than 20 years ago, when I was still young, handsome, and very promising. And I was working in St. Mary's Hospital in the center of London. And down just across where the Fleming lab, Alexander Fleming, discovered penicillin, was the UK first recurrent miscarriage clinic. It was a very large clinic. And one day I was asked to cover the clinic to provide clinical care. And I had no interest in miscarriages, like most of my colleagues. I thought, that is of no interest to me. But I had no choice. And the very first patient I saw was a lady who had come from the north of Scotland, so very far. And she had traveled because she presented with 8 consecutive pregnancy losses. Now, I was genuinely humbled because I didn't even realize that patients could have 8 consecutive. I also felt that I had not much to say to this patient. And at that time, they fill out a long questionnaire because people believe that lifestyle factors can cause it. And one of the questions is, how long did it take for you to conceive? And the idea was that if you're infertile and you become pregnant, whatever caused the infertility may also cause the miscarriage. And I looked at that questionnaire and said, how can you conceive in one cycle 8 times in a row? Now, the other reason why I put this slide up is because what we did in the clinic in 2003 is exactly what we're doing 20 years later. And it's exactly what we have been doing 20 years before I saw this patient. And the whole thinking around miscarriages is predicated on this idea that early pregnancy must be very vulnerable, very precious, and that a whole heap of subclinical disorders can cause miscarriages. So the standard approach is to do a lot of blood tests, whether it's for thrombophilia, metabolic disorders, immune disorders, etc. And the blunt truth is, report after authoritative report shows there is very little evidence or no evidence. Clinical trial after clinical trial comes up with no benefit of treating perceived subclinical disorders. As a matter of fact, there's even large population studies out from Norway where they looked at patients who actually have overt clinical disorders and found it doesn't impact miscarriage rate at all. And if you look at prospective studies, well, the only thing that correlates with future pregnancy outcome is maternal age, the number of previous miscarriages, and indeed non-surgical malformation of the uterus, of which in this cohort there were 43 patients in almost 2,000. So something is fundamentally wrong with how we think about miscarriages. And the way that we address this is to go back to basics and say, well, what does normal fertility looks like in humans? So imagine young, healthy women, imagine 100 conceptions. What happened to these concepti? Well, in healthy, normal reproductive women, at least 1 in 3 of these embryos will not make it to the implantation stage. They will falter before that. And from all the embryos they implanted, 1 in 3 will be lost after implantation. A majority so early that they will escape clinical detection, and that leave you between 12 to 15% miscarriage rate. 90% of them will occur in the first trimester of pregnancy before there are maternal vascular connections established with the placenta. And this prospective data is exactly the same across 4 large studies where they measured ultrasensitive ACG on urine in daily urine samples. And in many ways, we shouldn't be surprised that attrition rate in early pregnancy is so high, because we now know that human embryos are genetically unstable. So you have meiotic errors, they're rising in eggs, they will lead to complete aneuploidy of the embryo, and those are age-dependent. But even if you start with a healthy egg, it doesn't matter whether you're 45, 35, or 25, as soon as it fertilizes, there will be mitotic errors generated, leading to mosaic embryos. And the best estimate is that at least 99% of human embryos will have one cell that is chromosomal, one or more cells that is chromosomal abnormal. And humans are strange in other ways, because we are one of the very few mammals that actually have a menstrual cycle, which basically means that each conception starts with a genetically unique embryo implanting in a newly regenerated endometrium. And of course, human placentation is exceptionally deep. It's unrivaled amongst primates. And it's so deep, and it leads to such dramatic remodeling of the vascular arteries that literally the placenta can hijack the maternal physiology, exemplified by cardiac output rising from 45 mL per minute in the non-pregnant state to well over 700 in the 3rd trimester of pregnancy. So you look at this design and you say, this is incomprehensible, that the survival of the species depends on random embryos, an endometrium that keeps on cycling, and somehow that leads to immense investment in a very long pregnancy. And from a medical perspective, this is not quite understandable. There are no comparators in the body, but the evolution biologists have long pointed out that it may not be strange, because reproduction is under intense evolutionary pressure, but the pressure on the embryo is to maximize the chance of implantation, whereas the pressure on the mother is to evolve strategies to minimize prolonged investment in a failing pregnancy. So you have a tug of war. Now, how does this look like from the perspective of the endometrium? Well, the endometrium, I think, is seriously overlooked, seriously underappreciated. it is nothing close to spectacular. So it's the only tissue, I believe, that can quadruple in volume and thickness, literally from the end of a menstruation to before ovulation. That is 9 days. There's not even an aggressive cancer that can quadruple in 9 days. And that growth is spatially organized. You have morphogen gradients, cytokine gradients, which means that the cells in the endometrium during the growth get a positional identity, and you get stratification of that tissue. And after ovulation, progesterone will act on that, and uniquely menstruating species will trigger an acute inflammatory response that is called the decidual reaction. And once the decidual reaction is initiated, stroma cells, inflammatory stroma cells, will be transcriptionally reprogrammed into anti-inflammatory, highly stress-resistant cells, and you get accumulation of immunotolerant immune cells. So once it's initiated, it becomes critically dependent on progesterone. So if no conception happens, no ACG, the corpus luteum will falter, and you get menstruation. But of course, in case of pregnancy, that tissue will have to be transformed into a matrix, the decidua, that can literally anchor and support the placenta for 9 months in pregnancy. And by week 20, mid-trimester, it's only a few millimeters thick, and the placenta is massively bigger. One little thing to point out, what happens in no primates except for great apes, is that the embryo does not develop in the cavity in humans. The embryo will entirely implant. It's called interstitial implantation. It's the most recent evolution and innovation in the primate lineage. Now, for some years, I've pointed out, and there's a lot of work done, that this design is not as crazy as it may look like. Because once the decidual reaction is initiated, the tissue is in a bistable state. It can either menstruate or transform into a pregnancy tissue. And poor quality embryos invariably will disrupt this reaction, meaning that the endometrium will ensure that these embryos are actively eliminated. So in other words, the human endometrium is as much designed to get rid of embryos as it is to support high-quality embryos. And that is called the implantation checkpoint. And the implantation checkpoint is an important concept because it now decodes the event at the time of implantation. So for each conception, you can have an endometrium that is balanced. It'll allow an embryo to implant, but it also can transition into pregnancy tissue, or imbalanced, and each embryo will either be high fitness or low fitness. So if a high fitness embryo is implanted in a balanced endometrium, well, then you can expect you will have live births, unless you have certain chromosomal errors, like from small chromosomes, that literally don't impact early development in the embryo. They escape the implantation checkpoint. If a low fitness embryo implants, well, then you have physiological failure. So I also can't think of another system in the body that is designed to make it fail. So it's pretty unique to the uterus. If you have an endometrium where interstitial implantation cannot happen, well, then you have pathological failure. And if the endometrium cannot transition into a pregnancy tissue, well, it doesn't matter whether you're low or high fitness, you will end up with a miscarriage. So the whole question was, how can we test this? How can we prove that this is the case? Well, our tools are very limited because in the clinic we can do ultrasound scan, measure the growth. If you wish, you can measure progesterone levels, and there are some timing methods out there. But we can't measure whether the endometrium is ready for interstitial implantation, capable of transitioning into this decidual matrix, or has the right immune adaptation. So that is what we set out to develop tools that can measure this in clinical samples. And much of what I'm going to tell you was published this year in Science Advances. And the key discovery there was the discovery that human endometrium harbors a specialized interstitial implantation niche. This niche is populated by epithelial-mesenchymal stroma cells. They have a specialized ECM, and they really drive the interactions with the embryo. Now, importantly, one marker for this niche is DIA2. It's the enzyme that converts inactive thyroid hormone into active thyroid hormone. And a marker for the underlying inflammatory predecidual cells is PLA-G2A, which will generate arachidonic acid and stimulate the prostaglandin synthesis. Now, if you look at this immunofluorescent picture, so now DIA2 is in red, and PLA, the predecidual cells, are in green, you do not see yellow. And that is because the DIA2 cells are literally progesterone resistant, and the PLA-G2A cells, the predecidual cells, are strictly progesterone dependent, and a cell cannot be resistant and dependent at the same time. And what happened across the implantation window, in a time-sensitive manner, you go from a dominance of these cells that allow interstitial implantation to dominance of the cells that enables you to transform the endometrium in a seamless manner. The second event in the endometrium is this massive proliferation and accumulation of uterine natural killer cells. And again, you have cytolytic natural killer cells, which are just NCAM1 or CD56 positive. And once they proliferate, they will become KIR-positive cells, specialized immunotolerant cells. And again, there is a spatial organization to this. So the KIR-positive cells will colonize that implantation niche. So you have two time-dependent events across this implantation window, one in the stroma, one in the immune population. So what we did in order to start to quantify this, in order to define the physiological boundaries of this intrinsically inflammatory process, is to identify biomarkers for functionally distinct subsets and amplify them in a very large number of samples. So what we can do is then make the percentiles, or centralize the data for each day across the window when we take a biopsy. And because a tissue can have more glands, fewer, more stroma, etc., what we do to measure it, we make ratios of these biomarkers to take out that confounding factor. And then we literally can take an individual sample and start measuring in individual clinical samples. What is the strength of that decidual reaction? What is the strength of that expansion of these immune cells? So better viewed here as when we bin everything. And so you have, on the opposite, you can have a very weakened decidual reaction or extremely accelerated decidual reaction, and then K-cell expansion will just follow this. But while stroma cells are progesterone dependent directly, and K cells are not, they are uncoupled. So these are co-regulated events across the implantation window, and you see a lot of outliers. So the question is, can we use this in the clinic? And I'm putting up this slide. This is everything you need to know about miscarriages. It's come from Denmark, where the government has collected miscarriage data since the 1970s. So a country of 5 million inhabitants can generate epidemiological papers on 2.7 million pregnancy outcomes in 1.3 million women. And it's very clear that the probability of live birth rate will drop around the age of 35, which is a reflection that the miscarriage rate increases. Now, at the age of 35, your fertility also will drop, so it's double whammy. But there's a second driver, and that is independent of maternal age. Each miscarriage will increase the risk of a further miscarriage. And as I pointed out, often these patients present with superfertility, the opposite of infertility. And a few things, in the clinic, across the world, there is an arbitrary definition to decide what is normal and what is abnormal, based on a totally random number of miscarriages. there is no biological or epidemiological underpinning of this ridiculous, outdated approach. But more importantly, it also highlights something that is often overlooked. For instance, if you have 5 miscarriages, well, then you're still more likely to have a baby in pregnancy number 6 if you're under the age of 35. And clearly, any causal factor should increase in severity or frequency in line with the number of previous miscarriages. So this is what we set out to test in 3 distinct cohorts, a selection of patients just selected on the number of previous losses, nothing else, not age, not BMI, a prospective cohort, and we had samples from 2 cycles. And what we found, well, just to orientate you, on the left here is how often do you find a decidual reaction that is very weakened? On the right, how often do you find an accelerated decidual reaction? We found this, that the frequency of cycles where you have this blunting of the decidual reaction increased stepwise with each previous miscarriage, and you have a reciprocal decline in the heightened down. And NK cells, there's a trend, but it's not as clean. In the prospective cohort study, we found that if you find an abnormal decidual reaction in a random cycle, well, then you are increased risk of experience of future miscarriage. And when we take out the aneuploid pregnancies, their risk increases even further. In terms of natural killer cells, well, good expansion is associated, strong expansion with live birth, it favors live birth. So this really suggests that if you detect an abnormal decidual reaction, there must be a pathological state that recurs at much higher frequency than by chance alone. And that is exactly what we found. So if you look in the A biopsy, in the bins, and you ask yourself what happens in the B biopsy, well, both a stalling of the reaction or acceleration of the reaction occurs at much higher frequency by chance alone. Now, for the NK cell, they are extremely variable, so it's not quite clear, at least not significantly. So to explore that a little bit further, we looked at almost 3,000 biopsies where we determined the interquartile range of NK cells in the endometrium. And importantly, you can see that with each additional loss, that interquartile range becomes blunted, and that bluntness only starts at the 8 days after a positive ovulation there. Before that, you don't see any defects. After that is where you see the defects. So now we have these tools, and in the experimental reproductive medicine clinic, we have been using this tool for over a year. And the reason why we use these tools is to answer the question, if you experienced multiple miscarriages or multiple IVF failures, can we find objective evidence that the endometrium contributed to these abnormalities, to these failures? Because if we can, then we also have the opportunity to intervene before you conceive again. And a good example is progesterone. So the decidual reaction is progesterone dependent. And here, in about almost 1,000 patients that we've seen after developing the test, I'll plot the progesterone levels against the decidual reaction, all normalized for the day in the cycle. And it clearly shows that the process is progesterone dependent. But now you can identify patients who have an accelerated decidual reaction with very high progesterone levels. All of them are IVF patients. You have patients who have a very poor decidual reaction, but clearly have luteal phase progesterone levels, but very low. You have patients who have very high progesterone levels, also poor decidual reaction, so they're resistant. And then you have progesterone hypersensitivity. Not many patients, and I have a suspicion that they may be overrepresented by endometriosis patients rather than anything else. So, So the treatment becomes quite straightforward. If you have progesterone excess, ask the IVF doctor to calm down and lower their progesterone levels. You know, you can't believe how much progesterone some of these patients are. If you have progesterone deficiency, you give progesterone. But what do you do with progesterone resistance? And this goes back to previous studies where we showed that this blunting of the decidual reaction correlates perfectly with loss of clonogenic cells in the endometrium, loss of stem cells. Now, the endometrium is one of the few tissues where you can demonstrate beyond doubt that that tissue is dependent on cyclical recruitment of bone marrow-derived progenitor cells. And the signal that the endometrium uses is CXCL12, and that signal is inactivated by an enzyme called DPP4 in the blood, which inactivates a lot of peptide hormones, including hormones involved in glucose homeostasis. So we did a feasibility trial where we had patients randomized to either placebo or sitagliptin, and we found that after 3 cycles of sitagliptin, you had a significant increase in clonogenic cells, 14 more clonogenic cells per 3,000 stroma cells, while there was no effect in the placebo group. And more importantly, we found a massive response in enhancing this decidual reaction. So this is now still experimental, but the way that we treat patients who have bona fide progesterone resistance in there. So I'll leave you with this summary slide. You know, going from a cycling tissue into a pregnancy tissue is not a walk in the park. It's a highly time-sensitive process, and you go from a state where you enable interstitial implantation to a state where you have this predominance of decidual cells. If the whole process is accelerated, well, then you get pathological implantation failure. And if everything is stalled, well, then embryos find it that easy to implant. You know, you have super fertility, but the whole tissue will also break down in pregnancy and causing miscarriage. So finally, I'd like to thank the amazing team at Warwick, both the clinical team and the research lab. And on a final note, this lady from Scotland, in pregnancy number 9, she had a baby. Thank you.
Is there any question in the room? Nathalie.
So thank you very much for this very nice presentation, very interesting. I had a question concerning the last part of your talk where you mentioned the interest of DPP4 inhibitors. And as you mentioned, so this enzyme can cleavate a lot of peptides and so on. So how can you recapitulate which type of events, depending on DPP4, can explain this?
You're very, very right. So it was all based on the discovery, and quite compelling discovery from Hugh Taylor in Yale, that CXCL12 is a dominant signal to recruit these bone marrow-derived stem cells. And they will continue to be recruited in early pregnancy. And you know, I just looked up for what limits CXCL4, and there was DPP4. And of course, in the trial, we really show, although it was very limited in the number of patients, a very robust effect on clonogenic cells in the endometrium. So we just put 1 and 1 together and made 2 of it, or 3 of it. But you're very right. There's still doubt on what is the precise sequence of events that enable this. Yeah.
Thank you.
I have a question also. Do you think that the abnormalities that you see in decidualization is the cause or the consequence of the miscarriages? Because if it is the cause, how do you explain that it increases with the number of miscarriages?
It's a very insightful question, and you know, I think it's the consequence of miscarriage. So that miscarriages literally drive further miscarriages. So, and I have no way of nailing that, but modeling in assembloids really shows that prolonged decidual inflammation lead to stem cell depletion, which would be then the obvious way of connecting the dots. The important thing there is, of course, that there is no one in cardiology who would say, oh, have 3 heart attacks before we treat you. For the data set, if you want to have an impact, you should really treat after any miscarriage and identify those patients who are at risk of further miscarriages. So the data is not just academically, but it should lead to a much more informed and rational way of treating these patients. And honestly, it is generally true that miscarriage patients are very, very poorly treated across the world.
Thanks, Jan. That was absolutely spectacular. One of the bits of work you've done has been a really insightful work on senescence in stromal cells. And I wonder how the sort of senescence, aging, and other things that you've studied would fit into this new paradigm.
You see acute senescence, but in mid-luteal endometrium, there are just 4 sites of acute senescent cells, and they need to be cleared by activated NK cells, cytolytic NK cells. We in the past made slightly mistake when we thought that the DAI2 positive cells were senescent cells. And that is because senescence is associated with progesterone resistance, and in culture the emergence of DAI2 was a sign that we had cellular senescence. So we had to eat humble pie, hope that no one spotted our previous mistake. Some people did, which is good because they have read the previous papers. But yes, they are EMT cells, and they're definitely under the control of Wnt signaling from the lumen epithelium, these DAI2 positive cells. Yeah.
Thank you, Jan, and a really excellent, very interesting presentation. I just want to have a clinical question for a moment, and it reflects on the comment you had before Philippa's question about, the number of miscarriages, and you use the analogy of the number of heart attacks. So do you think in the clinical world we should no longer be separating the so-called spontaneous miscarriage from the so-called recurrent miscarriage?
Absolutely. So each patient, based on the Danish data, you can calculate their expected live birth rate and their expected miscarriage rate. So each patient you can give their heart information, and while each pregnancy comprises out of a unique embryo and a newly regenerated endometrium, and you can't give certainty, if you look at these predictions or these expectations at a population level, it's extremely accurate. So we did the exercise of looking at a cohort from Japan and calculate what the expected live birth would be based on individual predictions. And we were less than 0.5% out. So I think miscarriages are hardwired in biology. They're not a reflection. There are risk factors like BMI, etc. But deep down, they're just hardwired. And age drives meiotic errors and age-dependent risk, and it's the endometrium that drives this recurrence risk.
Thank you very much. Could you please tell us a little bit more about the function of those NK cells to induce tolerance?
So there are two aspects to that. So natural killer cells are in great abundance in pre-pregnancy endometrium. And so its function in pre-pregnancy endometrium is different to its function in pregnancy, where there's very, 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 very strong evidence that they are helping interacting with trophoblasts and actually are quite essential for normal placenta formation. They are by far the most variable cell type in the endometrium, in pre-pregnancy endometrium, throughout the cycle, but also between cycles. And that leads me to believe that they are critical homeostatic regulators of the endometrium. They're your helper cells that will sense when things are slightly out of balance and try to correct it. So we have some patients where we have, for instance, 6 biopsies of them. And you can see from very high natural cells, they go down over several cycles and they go up again. So rather than thinking about the endometrium in terms of one cycle, you should think about the endometrium in terms of hundreds of cycles across reproductive lifespan. And it's the resilience, how well that tissue maintains its resilience over hundreds of cycles that determines reproductive success versus reproductive failure. So it's a slightly different mindset that we need to adopt when it comes to the endometrium.
Thank you for your excellent presentation. Do you see difference in your fascinating observation according to the sex of the fetus?
So I can't answer the question. I think there are a few people in the world who still believe that. There's a particular researcher, Professor Christiansen in Denmark, who I never collected that data and I can't really answer it. It would be intriguing, but also like, what are you going to do about it, right? Type of.
So, thank you very much, Jan, for your brilliant lecture.
Merci, professeur. On me signale sur Internet que le son est assez faible. Est-ce qu'on peut le régler, le moduler? Ah, voilà, ok. Merci. Voilà, je ne savais pas. Merci beaucoup. Abnormal uterine bleeding, including HMB or heavy menstrual bleeding, impact at least 1 in 3 women of reproductive age. Professor Hilary Critchley is going to tell us how a recent clinical trial showed that a short-term dexamethasone treatment could improve HMB. Mr. President, she's also going to remind us how crucial cross-disciplinary collaboration is for new advances.
Professor Hilary Critchley is a clinician scientist and holds a personal chair in reproductive medicine at the University of Edinburgh. She has held senior university leadership roles as head of University of Edinburgh Deanery of Clinical Sciences and co-director in the Centre for Reproductive Health. She is an honorary consultant gynecologist at the Royal Infirmary, Edinburgh, since 1993. She is current chair of the UK Charity Wellbeing of Women Research Advisory Committee and past chair of FIGO Committee for Menstrual Disorders and Related Health Impacts. Her research program for over 30 years has focused on biology of the endometrium, disorders of menstruation, and abnormal uterine bleeding from biological and clinical perspectives. She is Fellow of the UK Academy of Medical Sciences and Fellow of the Royal Society of Edinburgh, and is an elected international member of US National Academy of Medicine. And she is also a world-known specialist for menstruation, and we will listen to her.
So, bonjour, good morning everyone. Professor Marbe, thank you very much, and the colleagues here at the Royal Academy of Medicine in Brussels. This is a real honour to talk in your presidential session, especially as throughout my career I have followed your incredible contributions to the field of endometrium and menstruation science, which, as has already been highlighted, the endometrium is far too long overlooked and so important. I'm going to switch from talking about implantation to what I consider its other equally important role, and that is of menstruation. So, what questions do we need to address when we consider abnormal uterine bleeding, which I'll refer to as AUB? Well, we need to understand the burden it imposes on those who suffer from the debilitating complaint. We need to know more about menstruation and its regulation and the causes of AUB. We need to move menstruation science forward, and we need to end the taboo on periods and period problems. AUB, in the context of global health, needs us to remember that 26% of the population across the world are women and girls of reproductive age. That means each month 1.8 billion are menstruating. And of that number, 1 in 3 at least experience abnormal uterine bleeding. Far too long we have had a one-size-fits-all approach to medical interventions. Those suffering are dissatisfied, and of those 1 in 3 with AUB, 1 in 3 will have a fertility-ending hysterectomy. Menstruation and its disorders also impose huge economic costs. In 2017, these data from the US on direct and indirect costs amounted to $37 billion annually. And even as recently as 2025, in the United Kingdom, the NHS Confederation published a report stating that £5 billion annually are lost to the UK economy due to absenteeism from work and presenteeism. That means women and girls are either at school or particularly at work in terms of this economic report, but are not working to their full capacity. Further, abnormal uterine bleeding, heavy menstrual bleeding, HMB, are major overlooked causes of iron deficiency and iron deficiency anemia. This very recent WHO report 2025 shows that anemia across the globe in reproductive aged women and girls is increasing, and the management of iron deficiency anemia and iron deficiency are not adequately addressed in guidelines for the management of HMB. And if we're considering anemia, this is the top of an iceberg, as far more are iron deficient. And menstruation should not be overlooked as we strive to control anemia. This slide is a take-home message about simultaneous management. I thank my colleague Mac Munro for the image. And what it shows that in those with abnormal and heavy menstrual bleeding, we may have therapies that stop the bleeding, they may or not treat the cause, but at the same time, we must replace iron. So it is a case of stop the bleeding, fill up the tank. Imagine you have a car engine running close to empty. Unless you fill up the tank, you will not sufficiently replace the losses. So let's consider the causes of abnormal uterine bleeding. And this image is a reflection of some 20-year journey. I had the privilege back in 2005 to co-chair with my colleagues Mac Munro from Los Angeles and the late Ian Fraser from Sydney, a journey about a process to improve the terminology and nomenclature for studying abnormal uterine bleeding. This has become known globally as FIGO System 1. And then FIGO System 2, which addresses the causes of abnormal uterine bleeding using the acronym PALM-COIN. And the structural causes include leiomyoma, and under the non-structural causes reflected by the acronym COIN, we have abnormal uterine bleeding of endometrial origin. That is bleeding when other causes have been excluded. And of course, within the structural causes, we have adenomyosis, and our final speaker will, of course, be addressing that condition. So menstruation, we've heard about its phenomenal capacity to support or not support implantation, but it is also an absolutely brilliant physiological example of a wound that heals repeatedly. It injures and repairs each month without scarring. And pivotal in that is the withdrawal of progesterone after sequential exposure to estradiol and progesterone, and then the removal of progesterone. But as we've already seen in the excellent previous talk, the endometrium is not simple. It is a multicellular organism, and these cellular components have to talk to each other, and at menstruation, these events are also exquisitely controlled, both in time and space, so that we can have this repeated sequence of proliferation, differentiation, shedding, regeneration, as many as 400 times across the reproductive life course. And this gives us plenty of time for those who menstruate to experience morbidity. I think clinical phenotyping is critical. And I do draw attention to this recent publication from Peter Rogers in Melbourne about molecular phenotyping and time as a continuous variable across the menstrual cycle. And if you look at the top panel A, what we see is that as we move through the cycle, and these are gene changes that at menstruation, prior to menstruation, but particularly in that circle, around that movement from the early to mid-secretory phase, just how rapidly the genes are changing. So we need to be sure that any difference we see is not due to the difference of response to steroid exposure, i.e. cycle phase, but they are real in terms of the underlying condition we are studying, and we need to get much better about our phenotyping. And beneath here are candidate genes and their respective gene changes over time across the cycle. I'm going to show you a cartoon now that captures a lot of work, both from our own group and others across the globe, that really summarizes what we know about menstruation, its underlying mechanisms, and in the context of menstrual bleeding. And this beaker shows a beaker with normal menstrual blood loss, and this beaker shows what would be considered heavy menstrual bleeding when we see not just exaggerated blood loss but prolonged blood loss. And what we know is that in response to the withdrawal of progesterone, we have again an inflammatory response. We've heard about the inflammatory and then anti-inflammatory architecture around implantation, but menstruation is an inflammatory event, not an infective event, an inflammatory event. And this is exaggerated when we have heavy bleeding. And pivotal also in the control of bleeding are these unique spiral arterioles within the endometrium. And normally with progesterone withdrawal, we have vasoconstriction of these in the response to generation of vasoconstrictors. But when we have heavy menstrual bleeding, and these are bodies of evidence from numerous groups, we have a less well-differentiated spiral arteriolar system. And we have an altered vasoconstrictor-vasodilator ratio. And so we have a slightly bigger radius. And think of all those multiple spiral arterioles that exist within the endometrium. And so if we could just have a small impact on each of those vessels and their radius, we would have a big impact on flow. So I want to move with that background to share briefly some data from 2 clinical trials, one a translational study and then another one targeting, I think, what will become the favorite hormone this morning, progesterone. So the first one I want to share is the DexFem study, and the second one, the Yukon study. So the DexFem study, funded by the Medical Research Council, and we are really talking here about team effort. It takes an army to deliver a clinical trial. But at the basis of this was the translational knowledge that women with heavy bleeding may have inadequate blood vessel constriction in their womb lining. And as I've just said, if we could modify that radius slightly of each of those vessels, we might have a big impact on blood loss. The study involved a short course in the luteal phase of giving a glucocorticoid, such that it might impact on the vasculature and inflammatory response. And the hypothesis was that glucocorticoids, we don't often think about glucocorticoids in the endometrium. They are also important, as well as estradiol and progesterone, but they do inhibit angiogenesis. And we've heard about one metabolizing enzyme. I'm going to introduce another one, 11-beta-HSD2, which normally inactivates cortisol. In women with heavy bleeding, we had already identified that it may be expressed in excess, and that would result in the endometrial tissue, a local glucocorticoid deficiency. And this might result in altered prostaglandin ratios, affect angiogenesis, impair vasoconstriction, and lead, or at least contribute to heavy bleeding. Dexamethasone was chosen as a glucocorticoid ligand because it resists inactivation to the metabolizing enzyme. And we've later showed that the luteal phase administration did in fact alter the endometrial steroid milieu. Our primary outcome was objective measured blood loss, and it was, we believe, probably one of the first in gynecology response-adaptive randomized placebo-controlled trials. And the bottom line was that at the higher doses, we did see an effect of dexamethasone reducing menstrual blood loss. I want to move now to the second study, which brings us back to progesterone. And we know progesterone withdrawal is pivotal in menstruation, and it is also an opportunity to again highlight the terrific contributions from our president, Professor Marbet, and his colleagues in this field, and the models and studies he has performed to contribute to our knowledge of menstrual regulation. This trial I want to talk about is targeting the progesterone receptor directly. And this is known as the UCON study. So this compared a progesterone receptor modulator, ulipristal acetate, with the most commonly used treatment for heavy bleeding, the levonorgestrel releasing IUS. And this was a head-to-head phase 3 clinical trial funded again by the Medical Research Council. And we used in this time a primary outcome which was a quality of life score. Importantly, the MHRA and the European Medicine agencies allow quality of life as a primary outcome in menstrual bleeding studies. And this tool measuring quality of life, the MIMA score, showed in both groups an improved quality of life. In those using ulipristal acetate, amenorrhea, that is absence of bleeding at 12 months, was more common in those using the selective progesterone receptor modulator. We had no problem with endometrial malignancy. We did see a small number with these unique class progesterone receptor modulator associated endometrial changes, PAEC. They resolved at 6 months, and importantly, no impact on the liver, no drug-induced liver injury. Within that clinical trial, this particular MRC scheme allows an embedded mechanism of action. And so we're very grateful to the patients who allowed themselves to be serially sampled before they started treatment, on treatment, and at the end of treatment. I just show you here histology of the normal endometrium, the secretory endometrium, and the very unusual phenotype we see when we use a progesterone receptor modulator. We have these large cystic glands. They are ringed by a single cuboidal layer of epithelium and exhibit a low, and I do emphasize, a low mitotic index. They also have a profound effect on progesterone-regulated endpoints. This is another one of these metabolizing enzymes that either activate or inactivate steroids within the endometrium. This is 17-beta-HSD2. It inactivates estradiol to estrone. A progesterone receptor modulator ablates its expression. And this could be then interpreted as creating a local estrogenic microenvironment, one you might think would drive proliferation. Importantly, these changes at a molecular and cellular level are reversible, but we are left with a very interesting paradox. we have a compound that drives an estrogenic microenvironment, yet we see a reduction in proliferation. This is a cell proliferation marker, Ki67, and no mitosis and no hyperplasia. And I think these compounds provide us with a really interesting tool for understanding better the action of progesterone on the endometrium. So, if we're going to look incisively, we have to have models, and there are many now who have used or are using a mouse model of menstruation. And I show here the simulated mouse model of menstruation. Our next speaker, my colleague Philippa Saunders, is in the audience here, together with Philippa and her PhD student Fiona Cousins and others in the teams. The Edinburgh team refined further the original model described by Finn and Pope and then later Brasted. And these mice menstruate because you remove ovaries, replace estradiol, then estradiol with progesterone. You've heard from Professor Brosens the importance of decidulization so that when you remove progesterone, the endometrium bleeds. And in this study here from Greg Armstrong, a previous PhD student with me, in very carefully phenotyped samples at the time of menstruation with progesterone withdrawal in the human, and then recapitulated with the model at the equivalent time points in the mouse, we can demonstrate that these important sequence of events leading up to menstruation, apoptosis followed by inflammatory cytokine influx and then neutrophil influx, can be recapitulated. We have huge gaps still in menstruation science, and I just want to, in the last few minutes, speak about two. One of them is the impact of endometrial aging, and the other one, and I don't want our last speaker to think I will tread on toes, I will not, but I do want to just talk about how we might better identify adenomyosis, and again, it leads me to have the opportunity of highlighting the contributions from our president and his excellent review on ectopic endometrium. So, the aging endometrium, the previous speaker, Jan, of course, has highlighted that, and we know that cellular compartments will age, or we believe age, because we see an impact on reproductive performance. Interestingly, this study, it was published this year, 1,000 people, half females in red, half males in blue, show again the differences in hormone exposure across the menstrual cycle, the change, of course, anticipated with aging. But what it tells us is that studying menstrual health is far from a straightforward exercise when we think of all the players we have to take into account. And this graphical abstract, published just some months ago, comes from a study, Miriam Hemberger and Wendy Dean in Calgary, Alex Tosolova, my previous PhD student, now postdoc with Wendy and Miriam, and a BBSRC grant shared with Miriam along with Canadian funders, shows in this model of studying uterine and endometrial aging in mice, both transcriptomically and epigenomically, that in the epithelium, as you age, that is probably one of the first places where you see evidence of endometrial aging, misregulation of genes, and particularly we highlighted one, FOXC1, and this is associated with a hyperproliferative phenotype. These observations in the mouse model were capitulated in a mouse endometrial organoid system, which takes me to the use of endometrial organoids and their broader utility in reproductive health. And this cartoon from a review by Margarita Turcu shows just how broadly these organoids are now being used. Margarita's contribution in 2017 really moved the field forward. Professor Brosens and myself were co-collaborators on that study. It has been important, but of course we don't just study organoids in isolation. We have to study them in their physiological context, side by side with the stromal cells, probably introducing the immune cells, the vasculature, the blood flow to make it live, and the matrix in which these cells grow. Finally, I want to just show the power of cross-discipline and cross-sector collaboration and return momentarily to our condition called adenomyosis, which in the context of abnormal uterine bleeding and heavy menstrual bleeding is a hugely under-recognized cause. And what we really need to be able to do is detect earlier. Here is the review from Camboni and our president, Professor Marbe. It shows ectopic endometrium within the myometrium, but of course, this is after a hysterectomy, and we absolutely need earlier diagnosis. And so the last set of data are some pictures from a feasibility study to ask, are there modes of detecting adenomyosis earlier? And this is the utility potential of magnetic resonance elastography. The study is led from Edinburgh by Varsha Jain and Neil Roberts. It is cross-discipline because it includes medical physicists, radiologists, gynecologists, mathematicians. It includes cross-institutional collaboration. Professor Dick Amon pioneered MR elastography at the Mayo Clinic, and they were essential partners in this study. And what this image here shows, so in these upper panels, what we're looking at is the uterus and wavelengths going through the uterus, and this is the sort of normal pattern of wavelength passage. In the bottom panels, again the uterus, but waves transverse much slower if there is fibrosis, and these color changes reflect where there may indeed be regions of excessive fibrosis. And we're very grateful to the patients in this study because this was done before they had their hysterectomy. So we were able to identify what we believe was the same region of interest, and this is the hysterectomy, and then histologically study that with markers of fibrosis. So this is the region of interest, and then this is a stain, for example, for collagen, the classic marker for fibrosis, which is at best a proof of concept study. Of course, further work needs to be done, and I show it again to just show how powerful cross-discipline, cross-sector collaboration is. So in concluding slides, I'd just like to share what I believe are challenges and opportunities in the field of abnormal uterine bleeding, which after all is a challenge in itself because we are talking about a symptom. We are not talking about a diagnosis, and it is much easier to study a diagnostic entity than it is a symptomatic complaint, and why we need to understand the cause. So we need new treatment approaches, much more personalized approaches, and better tools to assess. We have to acknowledge there are limitations of models. The phenotyping I cannot overestimate as the single most important factor as we develop further the potential of molecular characterization and interpretation of large data sets. We need to develop new approaches, and I've already raised the issue of these microphysiological systems, and my colleague, Professor Linda Griffith at MIT, is a real leader in the field here and brings cross-sector expertise to the field. And there is this power of collaboration, working as teams, cross-discipline and cross-sector. And this is a field far too long neglected. But in the wider context, let's call time on heavy and painful periods. Well-being of women, in February 2025, released this period symptom checker with a QR code. It's now being used in the UK by over 80,000 patients to help their conversations with their general practitioners to move forward management of a previously too much ignored complaint. And if we are going to end the taboo on periods and period problems, then we really need to have sufficient funding for the basic science. We need to work across these many disciplines, epidemiology, hematology, pediatrics, because we know the impacts of iron deficiency on pregnancy outcomes and even fetal neurodevelopment, gynecology and obstetrics. There are wonderful opportunities to intervene across the life course, identify, for example, the problem of bleeding, treat anemia and iron deficiency, and improve outcomes for mothers and their babies, and then involve, of course, our patient advocacy groups and our policy makers. In closing, a huge thank you to collaborators, local, national, international, and I've apologise to anyone I have omitted. None of this work is ever possible without funders, and I think the last but most important thank you goes to the patients who contribute and help us in our studies, and a big shout out and thank you for those patients who give up time and involvement so generously. So thank you again, Professor Marbe, and your colleagues here at the Royal Academy of Medicine in Brussels. A huge privilege to contribute to this session.
Thank you very much for your talk. Is there any question in the audience?
While I was sitting through your talk, Hilary, I was wondering whether the opposite problem also exists, where periods are just not strong enough or long enough to develop. And the reason why I pointed out is that there's a particular miscarriage phenotype called missed or silent miscarriages, which basically present with very early fetal growth restriction, fetal bradycardia, fetal demise, and then it can take days, if not weeks, before the actual miscarriage happen. And often these patients, it points to a lack of histotrophic nutrition, and I find clinically often these patients report periods that just 1 to 2 days, 1 sanitary towel per day, seem to be exceptionally light. So is there a reverse disorder to heavy periods?
So, Jan, thank you very much for that question. I guess because one inevitably, although one tries not to have a focus, you do have a focus. And the one where the complaint impacts quality of life is obviously heavy prolonged bleeding, and that has been a focus. I think you raise a really interesting question, and I think as we understand more about menstruation itself, then not only can we understand more about why it's heavy, but also why it may be short. And I suspect you and I have talked about this over the years together. You show data on whether decidualization is advanced or retarded. How interesting if I could have the menstruation histories of your patients in your recurrent miscarriage clinic. Because if I can say, it is a great tragedy that a clinical trial, and I hope that the industry and the regulators are listening, we ask about every other system of the body, but we do not ask about the reproductive system. We do not ask about pregnancy outcomes. We do not ask about periods. And I'm sure between us, we could create a very few questions that would begin to address that. And this was highlighted during the COVID pandemic and the use of COVID vaccines, complaints of heavy periods or altered periods, but nobody could answer it because those questions hadn't been asked in the what we call the CRFs, the clinical report forms.
question?
On a une question. Ah, mais d'abord, monsieur le secrétaire perpétuel, vous avez une question? Allez-y alors.
Thank you for your very interesting presentation. As education is very important for the prevention, it's important for a young woman to know if the bleeding is normal or not. And do you think that it's important, or that it would be important to make a screen of the population, very young population in our countries, to treat and to prevent this problem?
So thank you. A hugely important question. Interestingly, receiving, at least in my own country, both, well, across the United Kingdom, and I stress across the United Kingdom, those very discussions are very live at the moment. That example of the period symptom checker, it's not a tracker, it's a checker. It's a one-off questionnaire of a few simple questions that raises whether period experience might be within normal boundaries or might be heavy. Because if it is heavy, every one of those individuals is at risk of losing iron and iron deficiency and developing anemia. And again, across the world, Canada has been a leader in this. It is about, should we be, first of all, asking questions? We need education in schools about what is a normal period, what is a heavy period, and potentially those at risk, opportunistically checking, for example, ferritin levels to see if they are iron deficient. Because you may be anemic, but you are going to be much more iron deficient, and your hemoglobin may be normal, but you may still be iron deficient. And for too long, a normal hemoglobin has been the single check, and so iron deficiency has been missed. So you treat the periods, but the patient still feels dreadful because she's iron deficient. So yes, I think there's a big education in schools. We need to address curriculums. We need to make sure medical school curriculums adequately address these really important gynecological issues.
We have two academicians here waiting online. First one, we're going to give the floor for a question, is Professor Jean-Marie Rakic. Professor.
Yes, I just wanted to know if there is any causative link with pain and AUB. And my second question, if there is time, is there any place for local treatment of AUB with, for example, dexamethasone or anti-angiogenic drugs?
So thank you. Two questions, if I heard correctly. The first one is, so can I just re-emphasize that when you talk about AUB and pain, these are symptoms, they are not diagnoses. I'm looking at our last speaker who will talk about adenomyosis, and that is an excellent example of a cause of both very heavy bleeding and debilitating pain. In terms of local treatment, I think that we have to, ideally, before we introduce any new intervention, it should be evidence-based, ideally with a phase 3 trial. The data I showed on dexamethasone was really a proof of concept study. It was in a very selective group of patients, all who had bleeding of endometrial origin. And if there were other causes, they were noted in the sub-analyses. We would not recommend taking that further forward until there had been a phase 3 trial. That was an oral treatment, and to date we do not have any local delivery system. By that I presume you mean either via the vagina or via an intrauterine route. So thank you for your question, and I hope that answers.
Thank you, Professor. And we will have also a question from Professor Cédric Blancpain.
Thank you very much for your very interesting talk. You insist at the end of your presentation that more basic research would be needed for better understanding the menstruation and its pathophysiology. And so my question is, since mice model do not menstruate, what is, in your opinion, the best preclinical model to study menstruation and its disorder?
So thank you for your question. So the mouse model can be induced to menstruate. I hope that was clear from the description of simulating the human menstrual cycle and then withdrawing progesterone. I think in terms of what is the best preclinical model, this is where more basic science research is needed. We've already heard that we need more research to understand the roles of what is governing implantation. Equally, we need that for menstruation. I think there is a future in the microphysiological systems, but they have a long way to go. A lot of people are working in this area, and what we have to be able to do is recapitulate human physiology, and that means studying the cells together on the right matrix with exposure to the right media, probably with flow introduced as well. So we will get there. It's going to not be tomorrow, but I know many groups are working on it. And so I think that, you know, it is humans that are menstruating, and ultimately we should be developing human models. But until then, these other models do have their utility, but their usual utility is looking at a particular pathway or a particular intervention.
Thank you very much.
Thank you very much for your very nice lecture.
Benign but highly impactful uterine disorder, adenomyosis, is still insufficiently understood. Its etiology and its pathophysiological mechanism remain unclear. Together with her team, Professor Marie-Madeleine Dolmans has identified macrophages as key mediator in lesion development. She will present a promising experimental platform for mechanistic investigation and personalized therapies. Mr. President, you will now welcome this member of the company and colleague.
Yes, thank you, Marie-Madeleine Dolmans, for being with us today. She earned a medical degree from the Université Catholique de Louvain in Brussels in 2000 and a PhD degree on cryopreservation and transplantation of human ovarian tissue in 2006, and she collaborated for the first live birth after transplantation of human ovarian tissue in Belgium. She is gynecologist and divides her time equally between clinical and research activities because she is head of the gynecology research unit at the Université Catholique de Louvain. She is past president of the International Society for Fertility Preservation, and her research work focuses on ovarian transplantation, endometriosis, and adenomyosis. And she is full professor, specialty editor for fertility and sterility, editorial board member for RMB Online and other gynecological journals. And she will talk on adenomyosis today.
Thank you, Professor Critchley. It's an honor indeed to speak in the presidential conference today with my dear colleagues and renowned speakers. So thank you for this. So, dear President, ladies and gentlemen, and dear colleagues, it's my pleasure today to speak about adenomyosis. But what is adenomyosis? If I give you this as a definition, uterine adenomyosis, another enigmatic disease of our time, it won't help you a lot, but still it will give you some insights that we don't understand the pathology. The pathogenesis of adenomyosis is still poorly understood. So to give some cartoons, thank you for the jingle bells, almost Christmas. So to give you some cartoons, you see here a uterus with an ovary, and inside the uterus we have seen that there is the endometrium. So the endometrium is supposed to remain at its place, which is the endometrium. But, so this is the endometrium here. Utopic endometrium, you see the glands, and in green here the stroma. But some glands, they like to travel and they will go inside the myometrium. So they are supposed to remain inside these 2.5 millimeter of the endometrium place. But no, they go way far from their location and they go inside the myometrium, like you see here, and they will form these islands of ectopic endometrium. So that is adenomyosis. How can we diagnose adenomyosis? So not until so long ago, the gold standard of the diagnosis of adenomyosis was histology on hysterectomy species. But of course, and luckily nowadays we have better, and we have expert sonographists that can do really good diagnosis of adenomyosis, but also MRI is becoming a gold standard. And Bazot and Daray in Fertile Sterile, they published the different types of adenomyosis. So now you have understood that these glands go inside. You can imagine they go inside only at one place, forming an adenomyoma, but they can go inside diffusely and form diffuse adenomyosis. And here you have the example of an adenomyotic uterus. So this is a benign uterine disease with ectopic endometrial glands and stroma inside the myometrium. Symptoms, we have had a very nice talk on heavy menstrual bleeding. So this was in the causes, but it gives also intense pelvic pain and infertility. And I believe, to answer the question that we had before, I believe that the more heavy the menstruations are, the more painful they will be, because there is more efflux and the uterus needs to contract more to make all that going out. The prevalence in the gynecological clinic for adenomyosis is 20%. And as we said, the pathogenesis is still not yet fully understood. So this is one of my PhD students in Fertility Sterility 2018. Pathogenesis of the disease, is it invagination, is it metaplasia? So there are three big theories. You can see A, B, C. So A is invagination, B is Mullerian remnant metaplasia, and C, stem cells differentiation inside the myometrium. The most common theory is the invagination one, where you see these endometrial glands going inside the myometrium to form that ectopic endometrium, which is called adenomyosis. The origin of that endometrial invasiveness, well, that is the whole key. It is talked about local hyperestrogenism that provokes uterine hypercontractility that will provoke the injury to what we call the junctional zone, which is the yellow here that you see. And once you have that injury, well, you can have the endometrium that invades the myometrium through the disrupted junctional zone and cause the lesion, the ectopic lesion establishment. That's it so far. But what we know from our clinical data is that there is an association between uterine adenomyosis and infertility. It's associated with decreased rates of embryo implantation, lower clinical pregnancy rate, higher miscarriage rates, and increased risk of pregnancy complications. The endometrium from adenomyosis patients may be characterized by a displaced window of implantation, and we have seen that abbreviation in your slides, Jan, aberrant accumulation of inflammatory and immune cells, and decreased or asynchronous expression of markers involved in the receptivity of the blastocyst. So that is why, according to that, we led 2 investigations in my lab, which I will present you briefly today. So I will focus only on that. First investigation, endometrial receptivity in adenomyosis patients' endometrium. So that is a study on the endometrium itself. And when we speak about receptivity, so that is during the window of implantation, the trophrectoderm of the blastocyst will attach to the endometrium and subsequently invade the endometrial stroma and vasculature. So there are 3 phases during that blue period of window of implantation in mid-secretory phase. So the blastocyst will have the apposition, adhesion, and then invasion, as we have seen. And you see here a lot of markers that I will not tell the whole list today. Some in bold we studied in our lab, and I will pick up some of these just to share with you today. So we go to the study, expression of these endometrial receptivity markers in adenomyosis. So the study compared endometrium, endometrial tissue, from healthy controls and adenomyosis patients, all premenopausal without any hormonal treatment, diagnosis done by ultrasound or MRI, and of course, in the healthy group, no signs of adenomyosis nor endometriosis. Result number 1, progesterone receptor. We have talked about progesterone before, very important for the ongoing pregnancy. Well, you see that the progesterone receptor expression in healthy endometrium compared to adenomyotic endometrium, if we look at the stroma, it's significantly different in the secretory phase. You remember, window of implantation is about day 21 of the cycle. It's just the middle of the secretory phase. So that is a phase we are looking for if we speak about infertility and attachment of the embryo. And we see that in the secretory phase, well, it's significantly different. The amount of progesterone receptor is dramatically decreased in our patients with adenomyosis. And the leaf expression, which is also a marker of implantation of the embryo, is significantly decreased in the secretory phase of those patients with adenomyosis. And the last one I will present, so you spoke about Hugh Taylor in the US, who did a lot of work on the endometrium, and OXA10 is a really important marker for the implantation of the embryo. And you see the levels here of OXA10 in the patients with adenomyosis is dramatically decreased. And the last result I will show you is these pinopodes. So these are extrusion of the surface of the epithelial cells in the endometrium. And these are pinopodes that you see here. So that is what we see in histology. So we know that the surface, the epithelial cells, they present some cilia that we can recognize by histology. But if you have a look at a healthy subject in mid-secretory phase by electron microscopy, this is a picture that you can obtain. So let's have a look more deeply. So endometrium from a healthy subject, we can see ciliated cells that are shown at a bigger magnification with a red arrow. We see these pinopodes, so it's like the balloon effect. So these are normal in healthy patients mid-secretory phase. What about our adenomyotic patients? Mid-secretory phase. So this is one example of the electron microscopy of these endometrial biopsies in these patients. So we don't see the pinopodes, but we see regressed pinopodes that you see in blue here, and some rare ciliated cells still there, but it looks a bit like a desert there. The desert, not the desert. Desert is for later. Conclusion of that first study. Endometrium from adenomyotic patients is characterized by abnormal expression of receptivity markers. Stromal cells appear to be predominantly affected, showing the decreased expression of the progesterone receptor, LIF in the secretory phase, and much lower levels of OXA10 in both proliferative and secretory phases. This impaired endometrial receptivity and decidualization may contribute to the poor reproductive outcomes we see in clinics in those patients. These results have been published by Christina Stratopoulou, my PhD student. She has defended her thesis now. And I will go on. Hilary did a nice introduction on the topic. talking about Turco's pioneering work on organoids. We have Boretto also, who did pioneering work on organoids. And we wanted to mimic in vitro the disease. So the disease with endometrial assembloids taken from biopsies from adenomyotic patients. What is an assembloid? So we like to be in vitro because it's much easier to work there with the tissue. So how do we do an assembloid? Endometrial biopsies are taken. So again, thank you to the patients, but also this is a huge sampling work. So endometrial biopsies are taken and we separate by enzymatic dissociation the stromal cells and the epithelial cells. The problem is that we know that epithelial cells, they don't grow in 2D culture. You put them in vitro in your Petri dish, 1 day, 2 days, they are gone. They don't survive. So the pioneering work of the organoids was to make these epithelial cells survive in 3D cultures. So you have a bump of matrigel, and you put these epithelial cells there, and they will self-organize. They will self-assembly in glands. They will form 3D glands. And you see these glands here, glandular organoids. You recognize very well that they are doing, well, circles here, they are glands. And you recognize also the 2D Petri dish with just stromal cells, 1 layer there. It's good to have these 3D glands, but they don't represent really the endometrium, because we know both are very important, and the stroma and the glands. So the whole challenge was to put them together and to perform these assembloids, what we call. So this is put together the stromal cells and the glandular organoids. And in fact, they do survive in vitro. Day 0, you see these stromal cells, you recognize the glands here, 3D glands. Day 2, they do survive. And if we check the expression of the epithelial and stromal markers in those assembloids, well, you recognize the normal endometrium here. The stroma is CD10 positive. We have some stroma CD10 positive in the assembloid, and the glands are CK22 positive. The glands are CK22 positive in the assembloids. Of course, there is always place for improvement. Luckily, science is advancing. And if we can make a comment on that, is why are there so few stromal cells in the assembloids, for instance? So Chloé Bossard, another PhD student, is still ongoing, is trying to refine the endometrial assembloid model. And so, as I told you just a few minutes ago, the usual way to do organoids and assembloids is to have a matrigel drop, to have these 3D structures. And these are the viability tests. Well, at 10 days, you see that in the matrigel drop, it's more red, so more dead than viable. And if we put that matrigel drop upside down, we call it the hanging drop with an insert in the 3D culture system, well, the viability is really good until day 10. So going on with that structure, we will see how these assembloids will answer to the hormonal stimulation. We know there are 2 phases in the cycle, estrogenic phase with a proliferation, and then we have after ovulation progesteronic phase, secretory phase with progesterone. A woman cycles 28 days. We know in the mice it's 5 days. Well, in vitro it's rather 5 days also. So you do 2 days of estradiol, and then you go on with 3 days of progesterone, and then you obtain after progesterone stimulation what we hope a receptive endometrium. And let's have a look if that is the case. Well, unstimulated organoids, we have these stromal cells that are fibroblast shaped, the large endometrial glands with a thin epithelial layer and minimal gland secretion, that is without hormonal stimulation. And when we put the progesterone stimulation, well, we do have all the characteristics in vitro of receptivity. So naked nuclei-like appearance of these stromal cells, the smaller endometrial glands, but with round, basally located nuclei, and abundant gland secretion that you very well see here. So we go back to our patients with adenomyosis. And so we took endometrial tissue, so the biopsies from healthy control, N equals 6, and adenomyotic patients. And on all these patients, we did assembloids, so in vitro culture, and we tried to mimic the mid-secretory phases with the hormonal stimulation protocol. Of course, we had a look at the same markers I presented you in the first study, and we'll see how they respond in vitro. So first, leaf expression in those assembloids. So we'll first see how they respond in vitro. So we see that healthy assembloids without and with progesterone, we have that increase in leaf expression, so the marker of the progesteronic phase, secretory phase. So these assembloids do very well respond in vitro, and we see a huge difference in the leaf expression between the healthy patients and the adenomyotic samples. So again, Hoxaten, you remember, we see a very nice difference between the expression of Hoxaten in the assembloids coming from healthy patients compared to the assembloids coming from adenomyotic patients. And we will end with the spinopods. Can we replicate that receptivity in endometrium in vitro? So you recognize the same pictures as I showed you before on the tissue, healthy endometrial tissue and adenomyotic tissue. And what about assembloids? So this comes from the 3D cultures, and we see that if we have assembloids from healthy patients, we do reproduce these pinopods nicely. And if we have assembloids coming from a sample from an adenomyotic patient, well, it's the same desert with the pinopods and only these rare ciliated cells. So to conclude, these primary epithelial stromal assembloids can be generated from endometrial biopsies from healthy subjects and adenomyosis patients. The assembloids are responsive to hormonal stimulation. They do mimic the window of implantation by expressing all the receptivity markers and forming pinopods in vitro. Assembloids from adenomyosis patients show decreased immunostaining for leaf and Hoxaten and failed to physiologically develop pinopods. Patient-derived assembloids may serve as an advanced preclinical model of adenomyosis-related impaired endometrial receptivity, which can open new horizons in understanding and treating the condition. So this was recently published in Fertility and Sterility. But as a take-home message of this model of assembloids, so we would say that these epithelial stromal assembloids, they do replicate all the characteristics of the primary tissue. So if you have a look at genetic and epigenetic markers, they do replicate them very faithfully. They may serve as an advanced preclinical model to investigate the pathogenesis and also the therapeutic responses. Impaired endometrial receptivity, relying on stromal cell abnormalities, is characteristic of adenomyosis and may probably explain the poor reproductive outcomes we have in these patients. So endometrial organoids, we call it organoids, but as you know, with the stromal cells, it's rather assembloids. They represent really an opportunity for disease modeling application. And in the world, there are many teams trying to make organoids and assembloids from different pathologies, and they are used to have more insight into implantation failure, into adenomyosis, as like we do for the moment. And they do represent really a huge opportunity for therapeutic approaches with high throughput drug screening, because in vitro it is much easier to test all these drugs to see the effect on the pinopods, on the receptivity markers before going to clinical studies. So, of course, I want to thank you for your attention, and all this work is possible thanks to all my PhD students and colleagues in the gynecology research lab and my colleagues from the clinic, and as we know, from diverse sources of financing that are absolutely necessary. Thank you, everyone.
Marie-Madeleine. Are there questions in the audience? Jan.
Thank you. I really enjoyed the talk. And yes, we also been working for a very long time on organoids and assembloids and co-culturing human blastocysts with that. There's a technical question looking at your assembloids. Is this in matrigel? Are you culturing the stroma cells in matrigel?
Yes, so far we do.
So that is why you're stressing them out, because you can grow the glands as long as the stroma cells in hydrogel, in collagen hydrogel, and you will see that you have no cell death in your stroma compartment if you change the ECM. It's quite important. But the second question, or the second reflection, is that there is this, there's a new technique, new excitement about organoids, etc. But it also should be slightly moderated with a reality check, because, for instance, if the biomarker there use PLA-G2A, you will never, ever see it expressed in vitro, in primary cultures, 2D, 3D. And because when you culture cells, you will lose cell states. So the idea that these models faithfully recapitulate disease, is for...
I don't understand, because we have shown the biomarkers in vitro.
Yeah, no, but that is not... There are a number of selected genes that you can show.
Yes.
If you would do bulk RNA sequencing, you will have a subpopulation of genes that are aligned and a lot of genes that are just not aligned at all, that are in vitro. So I just want a little bit of caution, like patients with endometriosis, they have babies, a lot of them.
Surely,
so
luckily.
we have to explain why they can be successful if they're...
No, but the miscarriage rate is, well, according to the latest literature with the meta-analysis, there are more miscarriages than in the normal population.
Yeah.
I guess you do see them at your consultation.
Yes, but you also have to explain the successes. It cannot be that the endometrium is abnormal.
But if it's successful, we don't need to treat our patients. We don't see them. So, of course, as a medical doctor, as you said, we are focused on the pathology and the negative effect of it.
No, not quite what I intended, but yes, thank you. Yeah.
Marie-Madeleine, you showed that you have differences in the endometrial part of the tissue, suggesting that there is progesterone resistance in the endometrium of these adenomyotic patients. But don't you think that the myometrium is also important, and how could we analyze the myometrium?
Yes, there is just one article, it's not yet out, it's on Biorix, putting myometrial cells into the model. So, of course, we are far away from the endometrium because we lack myometrium below, we lack the endothelial cells, the vasculature, and we lack all the immune system that is of huge importance when we go for the next talk in endometriosis also. So we know that all these are implicated, and as Hilary said, well, in fact, we need to go to human. That's the best model, but it's not always that easy to get all these samples from these patients. So this gives us a model to study it a bit more easily, but so far myometrium is not yet in there, but it is the next step.
Marie-Madeleine, thank you very much for your informative presentation. I just would like to follow on from the question from our president. And in one of your early slides, you spoke about hyperestrogenism driving the contractility. So where is that coming from? Is it derived from the myometrium? Is it, if we're saying the endometrium is progesterone resistant, is that the consequence of that from the endometrium, or is it both?
I do like your question because I'm questioning myself since many years on that. it is in the literature for years and years, and we don't know. So Boulogne, they state that there is hyperhydroestrogenism because of the aromatase the aromatase that you have there, so the enzyme that is producing locally estrogen. But we never found aromatase inside the endometrium of hundreds of patients with endometriosis. And what we found was that the antibody that they used was not specific. So when you go to the Western blot, you don't find anything there. So that's a huge question. We still don't know, and I believe that was the reason of the title finally, is it is still poorly understood. And it's fine to say, okay, there is a disruption of the junctional zone, then the glands go inside. But why? And how does it come that there is that so-called contractility, so-called hyperestrogenism? I'm not 100% sure about the link between those two and between the cause of the pathology, to be honest.
No question on internet. So thank you very much for your nice presentation.
$190 million, that's the number of women affected by endometriosis worldwide. Current treatment options consist either of surgery or of drug that suppress ovarian hormones. Professor Philippa Saunders will now take us through a research to improve diagnostics and find non-hormonal therapies. Mr. President, over to you to introduce this panelist.
Yes, Philippa Saunders is Professor of Reproductive Services at the University of Edinburgh also, and co-director of the EXPECT Center, which brings together discovery scientists and clinicians conducting research to improve diagnosis and treatment of endometriosis. Her studies are currently focused on finding new strategies to reduce endometriosis-associated symptoms, including pain and fatigue. She is passionate about supporting career development in biomedical sciences and has trained and mentored more than 50 students and fellows. She is active on social media, highlighting the importance of women's health research, and has been recognized for her contributions to public engagement.
Thank you so much for your generous introduction, and I'd like to add my thanks to those of the other speakers for the great honor you've given us, inviting us to this lovely meeting. It's my pleasure here today to represent a large multidisciplinary team of researchers at the University of Edinburgh who are all focused on this very challenging condition, endometriosis. So, endometriosis is defined in our parlance by the presence of tissue lesions that resemble endometrium, but in the wrong place. We've been hearing about the endometrium when it's either within the endometrium doing its business and promoting fertility, or maybe invading the myometrial wall. In this case, we have small pieces of tissue that end up in other positions where they respond to hormonal signals and can bleed and become fibrotic. The usual site of these tissue lesions is within the pelvic cavity. So we have three main subtypes: the superficial lesions, which tend to be on the peritoneal wall, these ovarian cysts that form as large blood-filled structures within the ovaries, and the even more problematic deep lesions that can form profound areas of fibrosis and invade tissue. And it's interesting that quite often deep lesions will also appear in patients who also have adenomyosis. Somewhat more challenging for us is the fact that we can find these places of abnormal tissue in many other sites within the body, including in the lungs and in the diaphragm and the CNS. So this, as you've heard, is a very common condition and one that has exercised medical professionals for many years. The usual quote is that it might affect about 1 in 10 women of reproductive age. I think this is probably an underestimate because globally so much of the work has been done in first world countries, whereas in the third world underdiagnosis is a huge problem. It's profoundly debilitating for many patients. Work by ourselves and many others have shown that it can be hormone dependent, and we now tend to think of it as a neuroinflammatory disorder. So in the time I've been working in the endometriosis field with my clinical colleagues, I think we've gone from thinking very much in a lesion-centric way to thinking much more about the patient experience and the things that are bothering the patients, and how we as researchers and clinical professionals can address these needs, this unmet need in this very common condition. So the very puzzling thing about endometriosis is why the formation of what we think of as these small tissue lesions, which can often look quite small and quite indistinct on the pelvic wall, might be associated with so many different types of symptoms. Now, I'm sure we can just think that if you've got a big invading lesion and it's between the uterus and the bowel, you might expect there to be pain during defecation. But in fact, a lot of work has shown there's quite a poor association, particularly with these superficial lesions, between where they are and how many there are and how many symptoms an individual might have, particularly when it comes to pain, which is a complex sensory experience that can be heavily influenced by a number of different factors. So the guiding light of the work that we're doing in Edinburgh is really to take the whole complexity of this disorder and think about the individual's need at what stage in their life, and whether they want fertility or pain management or indeed other things that are bothering them. And one of the things that we've been studying a great deal more recently has been fatigue, which has come as a very problematic symptom for many people. So I'm happy to say that I think in the last 10 years or so, we've made big leaps forward in maybe understanding more about the complexity of the disorder. And this has come out of profound international efforts, led particularly from Australia and the University of Oxford in the UK, where we and others have pooled together the DNA samples we've taken from the patients, shared them internationally, compared them to those of other populations and other disorders. And there's been a real effort and indeed many insights that many of these multi-complex genetic risk factors associated with particularly these GWAS studies have shown up as being in common with a number of other disorders which share the similar symptoms. And I remember many years ago, we were having a meeting of patients, and there were 60 patients in the room, and I said, how many of you have got asthma? And half the women in the room put their hands up. And this was a moment, a sort of light bulb moment, that what we were looking at is a disorder that is a whole body disorder with many comorbidities. And we can learn a lot, maybe, from looking at other conditions. So as I say, we have, oops, gone too far, yes. What's going on here? Everyone's having a moment. Can I do anything? No, doesn't want to do it. It's okay. So, as I say, what we've done in Edinburgh is to think of ways that we can address these issues. And we started with surveying the patients. We then have developed a number of different systems, both in vitro and animal models, and then we've ultimately gone on to execute clinical trials. And at the center of this effort has been a big effort to gather as much information from the patients as we possibly can. And we've been very fortunate that we've been able to get samples, multiple samples from individuals. So we're not just taking a lesion, we're also getting the endometrium from that same patient, we're getting a bit of their peritoneal wall, we're isolating mesothelial cells, we're getting bloods, urine, saliva, feces, and also, importantly, this resource of peritoneal fluid, where we can look at the immune cell cohort within the pelvis. And by taking this information, these samples, but also all the clinical characteristics that the patients share with us, they fill in lots of different questionnaires, we have been able to build up many hundreds of samples with deep phenotyping. And we've done this using these protocols that have been internationally recognized, and that's allowed us to share samples across different places. So I'm going to share with you some examples of where these samples have informed the kind of research we've done and how they've led to clinical trials. So in this instance, we have used these assets and we've addressed some issues to do with what is a difference between a patient with symptomatic endometriosis and our patients are coming to us through pain clinics, chronic pain clinics. So they have symptomatic pain, they've been through the mill to get a diagnosis, and now they're at the pain clinic needing help. And we get samples from these people, and we are then able to analyze them. And doing this approach, we found a number of different pointers that have allowed us to maybe rethink what might be going on. But also, we are guided by the idea that what we want to do is improve therapeutic opportunities. So the first of the things that we observed was that there were really changes in the immune cell populations in the peritoneal fluid, but these correlated with the pain scores that the people had recorded. It didn't actually correlate as well with whether they had a lesion or not. So this is an inflammatory pelvic condition which may or may not have a lesion. Another observation made a number of years ago by a very talented PhD student working with Andrew Horn was that the peritoneal fluid, the actual fluid in the pelvis of the patients with endometriosis, had much higher concentrations of lactate. And lactate was made, it turned out, by, among other things, the mesothelial cells lining the peritoneal wall. So when we looked at the mesothelial cell behavior from the patients, they behave fundamentally differently to those from people who had pain but no lesion. And also that TGF-beta, also higher in the peritoneal fluid, was stimulating them to make the lactate. Now, you might think lactate's a bit dull and, you know, whatever, but we thought lactate was actually really very interesting because there's a lot of other data from other conditions that show lactate can promote cell proliferation and invasion. And one of the things we knew from our studies and those of others was that a lesion behaved as a proliferative entity and often invaded into other tissues. So we hypothesized that if we blocked this production of lactate, could we reduce the lesions, reduce their size, and maybe mitigate the symptoms, in other words, holy grail. Now, one of the strategies that we've adopted with limited funds and a guiding light that we want to get on with new treatments is to look at drug repurposing. So we go to the drug armamentarium and we go, what's out there that might help us mitigate this lactate being made? And we working in a UK context, are particularly interested in drugs that have been approved for use in UK patients. And one thing leapt out to us, and that was a drug called dichloroacetate. Now, this is a bit of a, well, if you mention dichloroacetate to pharmacologists, they kind of throw up their hands in horror and go, ooh, it's a horrible, dirty drug. But as a lead drug that had already been approved, it ticked all the things that we wanted. So it inhibited the enzyme that we had showed was overstimulated in our mesothelial cells. It had already been approved, and we were able to then look at the data on what doses have been used and to actually think about how we might test it. Now, to get money to do a clinical trial, we really needed to provide some preclinical data. And I've done a lot of preclinical data, and all models have their own negative caveats. But for this, we had two different models we were able to produce in order to convince the funders that we should be able to do the trial. And the first of these was a model we called a lesion in a dish. So, a lesion, in our parlance, was a piece of endometrium which had stromal cells, epithelial cells, immune cells, etc., which was on the peritoneal wall and associated with these mesothelial cells. So we took this to its most reduced version, and we had mesothelial cells in the base of the dish. We had medium, the so-called peritoneal fluid, bathing it, and in a co-culture system above it, in a transwell system, we put stromal cells. And the stromal cells are a major part of the lesion. We didn't have all the other cells there. So this is our lesion model, and we then ask the question, if we stimulate the mesothelial cells with the TGF-beta, do we get more lactate? And if we then put in the drug, can we mitigate this lactate production? And indeed, this turned out to be the case. In other words, it worked. the DCA reduced the amount of lactate, and the lactate reduced, reducing the lactate reduced the stromal cell proliferation as a surrogate for stimulating formation of a lesion. Now, the second preclinical model is a mouse model of endometriosis. Of course, this comes with all the caveats about mouse models. However, we used our menstrual mouse model, and then we did what we could do with this, which was we took the menstrual mouse material and we put it into the peritoneum of a recipient mouse. And this is to mimic the mechanism that's been much touted as being the source of endometriosis lesions, which is retrograde menstruation. So we are effectively giving retrograde menstruation to a mouse, and we get formation of lesions. We've shown that they phenocopy a lot of the typical things to do with a superficial peritoneal lesion, and they will respond to hormones, they develop nerves, they develop a vasculature. So in this model, we then used our DCA paradigm. We gave the mice endometriosis lesions, and then we said, if we look at the presence of the lesions, do we have lactate in the peritoneum? Yes. If we give the DCA, do we reduce it? Yes. Does this have an effect on the size of these induced lesions? Yes. Now, this was sufficient to convince the funders that they would give us money to do an initial feasibility study. So this was an open label, 30 women study, and we gave them oral DCA. They could have 2 different doses, and we were really looking to see whether the women found this acceptable. It wasn't powered to give us a big effect. We need a bigger trial for that. We tested the blood levels of DCA, and the paper has actually just come out. So interestingly, although this wasn't powered for a big effect, a lot of the women in the study actually said it made a big difference to their pain symptoms. This wildly exceeded our expectations, by the way. They reduced, and the metric of this was they reduced their existing pain medication. So we find this is a good way of saying, does something work? Did they reduce their pain meds? But another thing it showed up was side effects. So we knew this might happen. Some of the people got this peripheral neuropathy. They got like tingling fingers. It went away when they stopped taking the drug. But we did some other work to look at why this might be the case, and we found that there was a particular haplotype of an enzyme that would predispose you to be a slow metabolizer of DCA, so you got more higher levels in the blood. And this has informed a larger trial. It's been really difficult to get it off the ground because whilst DCA is out there, to get a placebo to it and also to get it at the grade that we can do it in a randomized controlled trial has proved to be extremely difficult. But we are going to do 100 women multi-center and for 12 weeks, and we're going to adjust their amounts of DCA depending on their genomic status. So this is, I feel, a major step forward in sort of a personalized medicine approach. I mean, anecdotally, many of the women who were on that initial feasibility trial were absolutely gutted that they had to give up taking the drug. They've been buying it off the internet and still taking it. So, you know, you can take that as you want. So we have heard mention of immune cells, and I want to emphasize that there's a huge body of work that shows macrophages particularly and other immune cell types may become dysregulated in this condition. A lot of the work ongoing is being led by Erin Greaves at the University of Warwick, where Jan is. Now, this is another clinical trial which is just starting. What Erin and Andrew Horn identified again was another drug that might target this particular phenotype, which was already in phase 3 clinical trials. And what Erin did was use that same mouse model that we developed in Edinburgh, which she took with her to Warwick, to test this drug and to see if it had an effect on the lesions, the number of sizes, and importantly, on a measure of pain response, which is of so-called von Frey abdominal response. And she showed the drug actually helped with this pain response. Pain is a very difficult thing to measure in rodents because obviously they can't articulate what they're feeling. Although they will squeak a bit if they get annoyed with you. And this is another trial that I'm really pleased to say, after much hassle, should start next year, and that's called MacEndo. So the other area that I've got personally very interested in is trying to think more holistically beyond just drug treatments and surgery. And this has again been driven by discussions with our local patient group who've expressed a lot of interest in other things they can do to help their own symptoms. They're extremely motivated to use interventions that might avoid them taking hormones. They're having to wait a long time for surgery, so they want to have things that they can use themselves. And this has actually become incredibly popular across the piece. There's been quite a few studies showing that exercise, yoga, and indeed physiotherapy can be extremely beneficial. The two areas we've started looking at particularly, again, very much motivated by patient feedback, has been cannabis products, but also dietary modification. Now, there's a long history of people using cannabis products, cannabinoids, for the treatment of chronic pain. They're widely used to mitigate pain in cancer, and in some countries they are indeed prescribed. However, after a number of different years, we've ended up setting up another trial, and this is being led by my colleague Lucy Whitaker, and this is going to be a 2-center, double-blind, randomized controlled trial with 100 patients. We are using CBT tinctures, so this has got CBD but not THC, because THC can make you tired and can give you more of the psycho effects. And this is going to focus mostly on pain and will be a bit about recruitment and retention. We've actually asked local patients about the use of CBD and cannabinoid products, and together with most international surveys and other, particularly in Australia, they've looked at this. Between 18 and 20% of patients do admit in a consultation that they are already trying these products. So we think there's really a need to get some more control into this area. And then the other area which has really taken off as an interest for me and others is looking at the gut microbiome. It's become a very sexy topic. We've been working in this area for a while. Ironically, my first degree was in microbiology, so after all these years, I've just come full circle back to looking at bacteria, which is something else. And in this concept, one of the things that's been so obvious to us is many of the conditions that are associated with endometriosis and comorbid with endometriosis are also those associated with gastrointestinal symptoms. And when we kicked off our PhD student project with Frankie Hearn-Yates, we first off did an international survey, which we promoted online for just 12 weeks. This is the biggest international survey that's ever happened. We had nearly 3,000 responses, of which 2,599 were complete. And in this obviously selected population, most of the individuals were suffering pelvic pain, but also a lot of them were also complaining of this condition called endobelly, where they get abdominal distension. They find it very distressing. It's much under-researched and needs a lot more attention. Obviously, this was, again, a population motivated to reply to a survey about diet and pain, and a lot of them had used the dietary intervention, and quite a lot of them had used a supplement. So we went on and did a pilot study where we took samples from ultimately 49 women were recruited. They did a lot of detailed questionnaires for us, including pain scores, the fatigue scores, and we had all their clinical data. And we took feces samples and did two different things. We did sequencing for the DNA, but importantly, we also did a metabolite extraction and a lot of bioinformatic analysis. And we received a great deal of support from colleagues at Imperial College in London, without whom we wouldn't have been able to do this study. And the DNA analysis was done by a colleague at the University of Cork in Southern Ireland. So once again, getting together with experts in other fields is really the great pleasure of these sorts of studies. This is a tiny snapshot of just one of the results we found. We found patterns of bugs, but also metabolites associated with pain, but the most strongest signal tended to be with fatigue, which I think is a very interesting one. And I just show you these unpublished data to emphasize there was a range of different metabolites, And in the model, it's really important one takes account of other characteristics such as BMI or hormone use or age, because all of these have an effect. We know age will have quite a profound effect. And one of the results we got from this was that of a series of different metabolites, those that were reduced were associated with a reduction in energy metabolism and production via the citric acid cycle, and we think this may explain fatigue. And in my last slide, I just want to press on with something that has been at the heart of the things that Andrew Horn and I have been trying to do for the last 5 years, which is to build up a much more holistic outline of what's going on in endometriosis. And one of the things that we want to do is longitudinal sampling in people, so we don't just take one sample, but we follow people through a different part of their life. This has just started recruitment, and I want to pay particular attention, credit to Mary-Madeleine, who's been involved in an EU grant that's allowed us to get started on recruiting the first 20 patients in the last month. EU Metriosis, there's going to be a tie-up with colleagues in Croatia, which is very exciting to be able to take people from a different culture. And another crucial person's in the audience here, my colleague Thanasis, who's our expert on data, and we've had some great times recently using phone apps and wearables to interrogate people. This is going to be the focus of our attention over the next 3 to 5 years, and we have managed to generate half of the £1 million that we wanted to have in order to launch it. Anyway, in closing, I will have undoubtedly left a few people off this slide, but I just want to emphasize none of this happens as an individual. It's all part of a multidisciplinary team. I'm deeply blessed to have so many great colleagues, particularly the patients who contribute, as well as the nurses who recruit them and the trial managers who undertake the trials. And it's been a great pleasure to work, particularly with my colleague, Professor Andrew Horn, over the last 12, I don't know how many years now, long time. Anyway, thank you for your attention.
Thank you very much. Are there questions in the audience? Nathalie.
So thank you very much for your very interesting talk and data. So I had a questions related to the last part of your talk, so concerning the microbiome story. So are there data relating also the vaginal microbiome with the different symptoms, and would it be also interesting to take it into account in parallel?
So we are going to be taking vaginal samples. We have not done work on the vaginal microbiome up to now. We came into this talking to people who work on what's called the gut-brain axis, particularly related to neurological decisions. And one of the leaders in the field is a guy called John Cryan, and he's shown that changes in the microbiome will influence your susceptibility to all kinds of conditions. Now, vaginal and endometrial microbiomes have been a subject of study by other groups. I guess for me, I'm very interested in the sort of pelvic relay, how the gut and its inflammation, how leaky gut, how might influence your whole body inflammatory state. So I'd sort of come into it from that direction. I guess I'm slightly less interested personally in the vaginal microbiome, but I do emphasize other people are looking at that, and people have found some interest. I think one thing to say about the microbiome is there's increasing numbers of studies, right, in endometriosis, but the groups of patients still, and ours was indeed the same, tend to be small. And a lot of these studies have no information about diet or exercise or hormonal status. I just reviewed a very interesting paper from China, and there they'd even taken fecal transfers into a mouse model. So there's a lot of energy, but I think what we need is an international consortium, a bit as we did with GWAS, where people go, this is what I found, this is what I found, this is what I found. Because I think if you read any one paper, they go, oh, this is the bug that's changed. But you know, I mean, maybe the diet's different, maybe they're younger, maybe they're taking, because we have seen a lot of an impact in a most recent study. We saw a lot of impact of how many painkillers they'd taken. And if you don't record these things, you don't know. And I think, again, I share Hilary's frustration. We're trying to do drug repurposing. We've got people coming to us and going, oh, try this drug. And I say, does it affect their menstrual cycle? We never recorded that. You just feel like sort of bashing a few heads together, to be honest. It's so stupid.
Fully agree with you.
It's just, if you allow me, Philippa, to complete the answer. As the leader, I believe I can say what Andrew is doing on the Endo 1000 that you presented. So Thanasis is involved. And in the Endometriosis EU project that I lead, so Andrew Horn, your colleague, is collecting vaginal swabs and feces. And they are doing, as you said, a huge set of patients in UK and in Croatia. So they are translating all the English stuff into Croatia. It's a very hard job to get all these home sampled. The patient do it themselves at home. I hope it's valid. And then they send it for analysis, and that's it. So there is interest indeed.
Yes, and it's an ambitious target. They've set themselves with at least 250 samples from multiple sites, including home-collected blood samples from patients in Edinburgh and an equal number in Croatia. And also, there's been a lot of work put into getting an app so the patient will record what their diet is. But importantly, also, we'll get information about the appearance of the stool at the time they took the sample, which is another piece of information that we need. So again, it's about building up this depth. And I agree, I mean, Mary Madeleine's done amazing work to get this off the ground, but it's very exciting to be able to do it across a couple of different sites. I think this is going to be one of the most powerful things. Do you not think it's going to be very good? It is going to be very good, and it's starting, just starting. Oh. Anyway.
Any further question?
Thank you for your very interesting presentation. I was really impressed by the amount of lactate that accumulate in this disease. And my question is, as this accumulation and this pressure, acid pressure, as an action on the general acid-base equilibrium of the patient?
Sorry?
On the general acid-basis equilibrium of the patient.
That's a very interesting point, actually. Yes, no, I don't think people are paying as much attention to that. There's been a huge amount on sort of profiling blood and all kinds of things, but that's a good point. I think, yeah, good point.
Because there are also very important connection between inflammation and general acid-basis equilibrium.
Yeah, no, absolutely. No, and I think these are all the kinds of things that we need people to be commenting and multidisciplinary approaches, because, you know, certainly I knew very little about pain mechanisms when I started working in the area, and I think we're still just learning, but you're absolutely right. Yes, good point.
Any question on internet?
Yes, Mr. President, we have a question from Professor Serge Goldman, who's going to connect. So, is this working? Maybe in the meantime. Yes, okay. He is there. Good morning, Professor Goldman. We can't hear you. You're on mute. You're on mute, Professor. You're on mute.
Okay. It's probably better. Do you hear me?
Yes.
Yes.
Okay, very good. First of all, I want to thank you for this very interesting call. And I wonder if you consider the role of the macrophage in the pathological process, considering this, if you have looked at the type of macrophages that we can find in the peritoneal fluid in those patients, I think about the M1 and M2 types and to see if there are a different differentiation between those two in those patients, with one with the pro-inflammatory and the other one with anti-inflammatory action. And also, I was also very interested by this increased lactate, as Professor Casimir was. And it's obviously something which is most probably related to the glycolysis and the glycolytic activity. And so I was wondering if you try to determine which type of lesions are responsible for this increase in the peritoneal fluid. And I was just wondering if, for instance, the pelvic, the deep lesions are more active to produce this lactate, or if it's more the superficial one, or if there is some differentiation between those two types. And I was just wondering if you have thought about using PET with FDG, fluorodeoxyglucose, that might evidence the glycolytic activity in any kind of local lesions to determine if we can differentiate the different type of lesions that might produce this lactate. Thank you.
Great, thank you for your question. I'll just take the last one first. We are doing a lot of work with PET-CT, looking at inflammatory markers, but also markers of fibrosis. It's being led by my colleague Lucy Whitaker, and we think this is going to be a breakthrough in diagnostic opportunity. So when we thinking about the macrophage work, there is a massive body of work, some of which was generated in our team, which has shown that there are changes in the subtype of macrophages, their phenotype, both within the peritoneal fluid, but also within the lesions themselves. And the lesions seem to represent a really unique sort of histological environment, which does seem to change the macrophage phenotype. And I think this is probably analogous to the situation in cancer, where the local niche within a cancer can change your immune cell phenotype. And a lot of this work is ongoing, particularly with Professor Greaves in Warwick. And yes, we see the macrophage as one of the pivotal cell types that might be open to some modification. In terms of the lactate work, we have only recently been doing much work on deep lesions. 70 to 80, we've got a pain clinic, and nearly everybody at that pain clinic has superficial lesions. So the work that was done to show that lactate change in the phenotype of the mesothelial cells lining the peritoneum was from individuals who had superficial disease. I can't answer the question regarding the deep disease because I don't have the evidence for that. At a genetic level, I believe the deep disease is actually has maybe a different risk factor, a different genetic origins. It's very much a fibrotic invasive disease. The superficial lesions seem to have a very different phenotype. I am, as a biologist, not subscribing to endometriosis being the same big bucket of things. That's why we focus on the symptoms. I hope that answers the questions, but thank you very much for the questions.
Thank you. Thank you.
And we also have, Professor, another question from the Young Academy. Professor Jonathan Doufis is here with us.
Thank you very much. Thank you for the very interesting presentation. I have one question regarding the exposure to estrogenic component and also progesterone. Do you think that you may have a different impact on the microbiome based on the type of estrogen for which the woman is exposed, and also to the type of progestin?
Oh yes, well thank you for that question. We definitely have looked at the hormonal status of the people from which we have the samples, and in the case of some of the bugs and metabolites, yes, you will see a change depending if they're on hormones or not. The other thing to say is there are subclasses of bugs within the gut that do metabolize estrogens, the so-called estrobolome. So these might be having a local effect. And we are taking all of that into account. I just really don't think we've got quite enough data at the moment to give you a definitive answer. But it does go back to my general comment that if people are doing these studies and they don't know the hormonal status of the people, they don't know if they're cycling and menstruating or on hormone suppressive therapy, they are going to get abnormal results. And we also need to know age and BMI. But thank you for the question.
Thank you.
Well, thank you very much, and congratulations for the very promising new treatment options.
Et nous en sommes au stade où vous pouvez envoyer vos demandes d'accréditation. Si vous nous regardez en ligne, et vous êtes assez nombreux aujourd'hui, Monsieur le Président, on avait le double de l'Assemblée présente ici en ligne, même plus du double. Donc je rappelle l'adresse que vous connaissez déjà tous par cœur, néanmoins je le fais, contact@armb.be, nom, prénom, numéro, INAMI. Et cela vaut aussi pour ceux qui ne sont pas membres de l'Académie, évidemment, et qui nous regardent sur YouTube. Et c'est ici que s'achève la retransmission, justement, sur YouTube. Merci de nous avoir suivis au fil des séances tout au long de cette année 2025. Rendez-vous le 24 janvier pour l'accueil des nouveaux membres belges et étrangers. Et d'ici là, passez de bonnes fêtes, prenez soin de vous et prenez soin de vos proches. On va donc couper YouTube. Et Monsieur le secrétaire perpétuel, pour les membres de la compagnie, vous allez maintenant nous faire part des communications de cette fin d'année.